All right, y'all, we're going to go ahead and start with chapter 2, part 2. If you're following along on the slides, you're going to be going to page or chapter 30. Slide 30 is the one we're going to be starting on. Okay. All right. So what are the functions of the various cerebral cortex regions? So the cerebral cortex, we've mentioned it a couple of times. So this part up here, cerebral cortex, is an intricate fabric of interconnected neural cells covering the cerebral hemispheres. So covering both hemispheres. The cerebral cortex is the body's ultimate control and information processing center. So we have the cortex and its basic divisions, right? So the brain has the left and right hemisphere, and then it has 4 lobes. Each hemisphere has 4 lobes. The frontal lobe, which is the front part of our brain, right here, the parietal lobe, which is this back, like the top of the skull to the back parietal lobe, the occipital lobe, which is right on the back of our skull, and then the temporal lobe, which is sort of in between our ears. So you can do that to remember the 4 lobes. The frontal lobe lies just behind the forehead, and it's involved in speaking, muscle movement, making plans, and judgment. It's also known as the executive center of our brain. The parietal lobe lies at the top of the head towards the back. It receives sensory input from touch and body position. The occipital lobe, which is the very back of our head, includes areas that receive information from our visual fields. So occipital, optical, right? Our eyes, that's where that information goes. And then the temporal lobes lie roughly above the ears and include auditory areas, receiving information primarily from the opposite ear. So sensory functions, we have the somatosensory cortex. It's the area in the front of the parietal lobe that registers and processes information from skin senses and the body parts movement. The visual cortex of the occipital lobe at the rear of the brain perceives input from your eyes, and the auditory cortex in the temporal lobes above your ears receives information from your ears. So somatosensory, it's the skin and the body. Visual, it's from the eyes, and auditory from the ears, right? So we have the visual cortex back here and that auditory cortex right there. Together, the small cortical areas that either receive sensory input or direct muscular output occupy about 1/4 of the human brain's thin, wrinkled cover. The remaining vast regions of the cortex are association areas. So association areas are found in all 4 lobes. The prefrontal cortex is the forward part of the frontal lobe, enables judgment, planning, processing, and new memories. And damage to association areas results in different losses. So we're going to look at an example of one case, and this is the case of Phineas Gage. So this right here is the person, Phineas Gage, and this is a illustration, representation of an accident that happened to him. So Phineas Gage's skull was kept as a medical record using measurements and modern neuroimaging techniques. Researchers have reconstructed the probable path of the rod through Gage's brain. So Phineas Gage was a railroad worker who ended up in a terrible accident, and he ended up with a railroad spike, as you can see, coming under the cheek, through the eye, breaking this cheekbone, and this bone right here going up through the frontal cortex, the frontal lobe. And he survived, which was really fascinating. And from that, we have a case study of what happens when someone has really, really traumatic damage to the frontal cortex. And so Phineas Gage, before his accident, was described as this very mild-mannered, very nice gentleman, right? Who was very kind, to go to his job. And then he had this accident and his personality completely changed. And he became this very like crass and rude man. Started taking up, I think it was smoking, when he previously was never a smoker. So it's a case study that can really give us some insight into really what all could be changed by impacting that frontal lobe, and the fact that you can survive it. This is a case study because it would be unethical for us to replicate this in an experimental setting. So we have to just look at previous cases like this to give us an insight on what happened and what are the implications of that. But to what extent can a damaged brain recognize itself, and what is neurogenesis? So if one hemisphere is damaged early in life, the other may assume functions by reorganizing and building new pathways. This is plasticity. So plasticity is the brain's ability to change, and this is especially during childhood. The brain reorganizes after damage or by building new pathways based on experiences. And this ability is diminished later in life. So if you are unfortunate enough to have a brain injury, it's really beneficial to have brain injuries younger, particularly in children. They have much higher recovery rate than, say, having a brain injury later in life, because of the plasticity of the brain in childhood that is not present in later adulthood. And neurogenesis is just the formation of new neurons. So what do split brains reveal about the functions of our two brain hemispheres? So lateralization, our left and right hemispheres serve different functions. This specialization is apparent after brain damage. So damage to the left hemisphere will impair reading, writing, speaking, arithmetic, reasoning, and understanding. Similar damage to the right hemisphere has less visibly dramatic effects, but researchers have found, that this minor right hemisphere is not as limited as previously believed. So damage to the left hemisphere is going to be much more apparent than damage to the right hemisphere, but that does not mean that damage to the right hemisphere does not have consequences. So this large band right here, the corpus callosum of neural fibers, connects the two brain hemispheres. So to photograph this half brain above at left, a surgeon separated the hemispheres by cutting through the corpus callosum and the lower brain regions. And then the high resolution diffusion spectrum image to the right here shows a top facing brain from above and reveals the brain's neural network within the two hemispheres and the corpus callosum neural bridge between them, right? So this bridge right here, that is the corpus callosum. It connects our hemispheres. So our lower brain regions, right, that cerebellum, the brainstem, those are all fused together. They're not separated, but our hemispheres, the upper parts, are separated. So what are chromosomes, DNA, genes, and the human genome? And how do behavior genetics explain our individual differences? So we talked a little bit about evolutionary psychology and genetics in chapter 1. We're going to go in a little more detail here. The environment, right, is every non-genetic influence from prenatal nutrition to the people and the things that are around us, right? That's the environment that we live in. Heredity is the genetic transfer of characteristics from parents to offspring. So things passed down. Behavior genetics is the study of relative power and limits of genetic and environmental influences on behavior. So we have chromosomes. Those are thread-like structures made of DNA molecules that contain genes, which is sort of like our genes are, what's a good metaphor for this? Are the ways, are what is used to create our physical expression, right? So the color of your hair, the color of your eyes, the size of your nose, right? All of these things are because certain genes are turned on or off. And chromosomes are part of that. DNA is a complex molecule containing the genetic information that makes up the chromosomes, the genes, those biochemical units of heredity that make up chromosomes. There's segments of DNA capable of synthesizing proteins. We have the genome, which is all the genetic material in an organism's chromosomes. When genes are expressed, they provide the code for creating the proteins that form our body's building blocks, right? So that's a good comparison. The genes are like the code of our body. When those codes are expressed or when those codes are written out, you see them in our physical makeup. And when they're turned off, you don't see them. And remember, we talked a little bit about epigenetics and how our environment can turn on or off certain genes. How do twin and adoption studies help us understand the effects and interaction of nature and nurture, right? So we have identical versus fraternal twins, and knowing the difference between identical and fraternal twins in studies tells us a lot about the nature versus nurture debate. Identical twins develop from a single fertilized egg that splits into two, and this creates two genetically identical organisms. On the genetic level, those two, that egg that splits is the same thing, right? Genetically identical. Identical twins share the same conception and uterus, and usually the same birth date and cultural history. You then have fraternal twins, and these develop from separate fertilized eggs. So as identical twins, it's one egg that splits. Fraternal twins, it's two eggs that were present and fertilized. And they're genetically no closer than brothers and sisters. And but they share a prenatal environment. So they share a womb, but they're essentially like having a brother or a sister, not the same as having an identical twin. Studies of twins in adulthood show that identical twins are far more alike than fraternal twins in like having disorders such as autism spectrum disorder, personality traits such as extroversion or neuroticism. If you're an identical twin, you're more likely to share those characteristics than being a fraternal twin. So separated twins, similarities found in identical twins, despite being raised in different homes, include personality, ways of thinking and relating to others, abilities and intelligence test scores, their general attitudes and temperament, their interests and their tastes, any specific fears that they have, their brain waves, and even their heart rate can be found to be similar, even if they are raised apart. So biological versus adopted relatives. Studies conducted with adoptive children whom the biological relatives are known. The adopted children seem to be more similar to their genetic relatives than their adoptive relatives. The environment shared by the family's children has virtually no discernible impact on their personalities. So does the home environment have an impact? So despite strong impacts of genetics on personality, parenting does have an influence. It has an influence on religious beliefs, values, manners, general attitude and temperament, politics, and their habits, right? And our habits are a huge one, right? The environment that we were raised in impacts our habits that can impact things like epigenetics. If you are coming from a family who exercises together like every day, you are generally going to be very fit. You're not, generally speaking, you might not have a lot of health issues if you're not, predetermined to have those based on your genetics, as opposed to being in a family who does not exercise or has McDonald's for every meal, right? You might be more likely to experience negative health outcomes because of those habits you learned in your environment. So how do heredity and environment work together? Genes and environment, nature and nurture work together. So genes are self-regulated rather than acting as blueprints that lead to the same results no matter the context. Genes react to our environments. So genes experience and interact with the environment and world that we live in. I've said this a couple times, epigenetics is the study of the environmental influences on gene expression that occur without any changes happening to our DNA. So epigenetics influence gene expression. Life experiences begin in the womb, lay down epigenetic markers, often organic methyl molecules that can affect the expression of any gene in the associated DNA segment. So prenatal, so this is an example, prenatal. You are exposed to drugs, toxins, some nutrition, and general stress in the womb. Then in postnatal, which is after birth, you experience neglect and abuse and variations in care throughout your childhood. Then as a juvenile, you start having more social contact and your environment gets more complex. And then as adult, you experience some cognitive challenges. You start exercising more and you start focusing on your nutrition. Each of these things has an impact on the genes and how those genes are expressed. So gene expression affected by epigenetic molecules. Evolutionary psychology is the study of evolution of behavior and the mind using principles of natural selection. Evolutionary psychologists use Charles Darwin's principles of natural selection to understand the roots of behavior and mental processes. Natural selection is the principle that those chance inherited traits are better enabled an organism to survive and reproduce in a particular environment will most likely be passed on to succeeding generations. Nature has indeed selected advantageous variations from the new gene combinations produced at each human conception, plus mutations, which are random errors that just kind of happen when genes replicate that sometimes result. But especially when humans, genes, and experience together wire the brain. So natural selection versus adaptation. Genes selected during our ancestral history give us a greater capacity to learn and therefore adapt to life in varied environments. So whereas we might have been in communities that could only really live in a certain area because that's, what their bodies were used to. And then as we become more spread out, our bodies change and adapt to fit the environments that we live in. The tight genetic leash that predisposes a dog's retrieving, a cat pouncing, or a bird's nesting is looser in humans. So we have more of a genetic variety in the way we interact with our world and our environment than, say, a cat or a dog has a very specific amount of behaviors and things that they will do in their environment, regardless of where they are. Adaptive flexibility in responding to different environments contributes to the ability to survive and reproduce. So how natural selection works, we begin with the species genome, which contains a variety of versions of a gene that shape traits. conditions make it difficult for individuals with some traits, some versions of those genes, to survive long enough to reproduce. Other individuals thus have their traits and genes selected to spread in the population because their genes allowed them to get to reproduction age and continue those genes to be passed down. An example of this in the wild would be if you have an environment. So we're talking about rabbits, and we are in like a foresty area. There's a lot of greens and browns and tans, like mud colors. And we have rabbits that vary in shades from all white to brownish to black. You're probably going to see after a couple generations, there's not a lot of white rabbits left. There may not even be a lot of black rabbits left because those are easier to spot out on the forest floor than a brown rabbit. So those traits, those genes that made their fur color white or black would be sort of cut out because they're not living to reproduce. Only the brown rabbits are able to reproduce because their traits allow them to live in that environment better. Same if we took these same rabbits and put them in a like a snowy area where it's continual snow. The white rabbits would be fine and they would continue to reproduce, and there would be lots of white rabbits, but the black rabbits or brown rabbits probably wouldn't be around because they're easier to pick out and predators would catch them before they caught the white rabbits. So as we go through different generations, if a certain trait is not making it to reproduction age, it very well may die out. So our genetic legacy. As success-enhancing genes were selected over time, behavioral tendencies and thinking and learning capacities emerged that prepared our Stone Age ancestors to survive, reproduce, and send their genes into the future. As inheritors of this prehistoric genetic legacy, we are genetically predisposed to behave in ways that promote our ancestors surviving and reproducing, ways that may be mismatched with our contemporary lifestyle. So an example is the way we like our food. Our prehistoric ancestors really needed their food to last a long time, right? They were only able to eat every once in a while, right? They had to really work to get their food. They were, hunters and gatherers. And one meal, right, one hunt maybe only lasted them a couple of days, and then it might have been a couple of days to a week before they had their next meal. So they needed their food to go a long time, which for our bodies means we're really good at holding on to like sugars and fats, because that's what their body needed to hold on to last them into the next hunt, the next gather, the next meal. Right now, for some people in some areas of the world, food is much more easily accessible. So we're eating 3 times a day, and some people more than that, and it's impacting our health. And we see higher rates of things like diabetes, cardiovascular disease, obesity, because the way our body is storing and holding on to the sugars and the fats and the things that we eat, and the fact that our diets are so saturated in sugars and fats and salts, is severely impacting our health. But from an evolutionary standpoint, we crave those kind of foods because that's the kind of food that would keep our ancestors alive. So there's that mismatch on how our bodies are sort of prehistorically disposed to do certain things, but they don't match the current lifestyles we have. So evolutionary psychology today, Darwin's theory of evolution has become one of biology's organizing principles. The theory lies on the theory lives on in the second Darwinian revolution, the application of evolutionary principles to psychology. Darwin anticipated this, foreseeing open fields for far more important research. Psychology will be based on a new foundation. Okay, and so we're going to stop there. That is the end of our chapter. If I can stop sharing my screen. There we go. So that is a lot of really dense information. There's the parts of the brain, the different systems, particularly the endocrine system, the central nervous system, the all of the different nervous systems. There's a bunch. A lot of definitions, a lot of graphs and charts, and it's a lot of information. Please watch this multiple times. I think I might also post Dr. Anderson's lecture. So if this one was really hard for you to grasp, you can also watch theirs. So you can see the information. Maybe there's a way I'm explaining something that Dr. Anderson explains in a different way that helps you grasp the material. But this is a difficult section. It's a difficult chapter. It is very dense. Please revisit this multiple times. Go through the learning curves multiple times. Again, they're going to be reopened before your next unit exam, so you can use it to study. When I start making the study guides for the unit exam, I will specifically go through and highlight the sections of this chapter that you need to focus on. It's a lot of material. Not all of this material is going to be on the exam. I would very much focus on the nervous systems. I would focus on neurons and that path of how information goes through a neuron. I would really focus on the genetics and epigenetics and how our environment impacts our genes and how our genes impact the way we work in our environments. But again, that's going to be on a study guide, so you know exactly what you need to be looking for. Again, go through the handout, go through the outline. I will probably post, maybe not this week. I probably will post an office hour within the next couple of weeks to try to go through some information. You can come if you have any questions, especially to prepare for the next exam, because this is a dense chapter. This next exam might be a little difficult if you're not really following along or grasping the material. So if you have any questions, please reach out. Another class update that I did not give on the front end, grades are starting to be posted. My goal is to have everything put into the grade book. I have a lot of grades on my own personal grade book, putting things onto the main grade book so you can see them, and then also send out emails that let you know where you are in the class as far as like your grade wise, if you're missing anything, so you can know by the October 14th deadline if you want to drop this class or not. The October 14th, that's the deadline to drop with, I believe, a W on your transcripts, or which is a withdrawal. And I do not believe that impacts your GPA. So my goal is to get everything graded, put into the grade book where you can see it, and send out individual emails where you can see what your grade is in this class and if you're missing anything. So that's going to be coming up in the next couple of weeks. Again, go through this material, watch this lecture a couple of times, go through the handouts, go through those learning curves, take notes. And then again, if you have any questions, please feel free to reach out to me or Dr. Anderson. Y'all have a great day.